Good day, dear students. In this module, we will be discussing about crossing over and sex-linked inheritance. Now, the first and foremost thing, what is crossing over? Crossing over is a recombination of genes that occurs due to exchange of segments between non-sister chromatids. Now, if you can recollect, we have studied crossing over last year in prophase 1 of meiosis 1, which included the following events, that is synapsis, tetrad formation, crossing over and terminalization. Now, we would be discussing about synapsis. Now, synapsis occurs in the zygotine stage of prophase 1. Here, the homologous chromosomes are going to form bivalence. So, this phenomena or this stage is called as synapsis. The second phenomena is crossing over. Now, what is crossing over? Crossing over takes place in the Purkinje stage of prophase 1. In this stage, as you can see, the two chromosomes are exchanging the genetic material and they are getting separated from each other. So first, they combine and exchange the genetic material, that is recombination. The combination or the point at which they are attached is broken down with the help of an enzyme that is called as endonuclease. The point at which they are attached is known as chiasma. So there are three events that are taking place. The first event is they come close together, exchange the genetic segments, that is recombination. The point at which they are attached is broken due to the enzyme endonuclease, and the point at which they were attached is known as chiasma, also known as chiasmata. Now, sex-linked inheritance. Now, sex-linked inheritance is due to the sex chromosome in a region that is called as the non-homologous region. If you can recollect, these are those regions which do not participate in crossing over during meiosis. Now, these regions are not involved in sex determination. These genes are called as sex-linked genes. They are responsible for inheriting sex-linked characters, and the pattern is known as sex-linked inheritance or sex linkage. Let's study sex linkage types in detail. Now, sex-linked genes which are present are of two types. One is the X-linked genes that is present on the X chromosome. These are also known as the sex-linked genes. And the second is the Y-linked, or it is also known as Hollandric genes, which are present on the Y chromosome. Now, the X-linked genes are present on the non-homologous region of the X chromosome. So these genes are called as X-linked. These are also known as sex-linked genes, and no corresponding allele of it is present on the Y chromosome. Now, examples for the same is short-sightedness, also known as myopia, a bleeding disorder that is known as hemophilia, color blindness where the person cannot differentiate between red and green colors, then night blindness where person does not have a night vision, and the last one is a serious disorder that is muscular dystrophy where the skeletal muscles of the person start degenerating. Now, female contains a double dose of X chromosome, that is X and X. So in most cases, she does not suffer from X-linked diseases which are there. When a female contains one recessive copy, she will become a carrier. If she contains two recessive copy, only then she will be diseased. But a disease state in case of female is a rare condition that is seen. In most cases, she contains one faulty copy and acts as a carrier. In case of male, it contains only one X chromosome which is there. So if one of the X chromosome that is there is faulty, at that time it is diseased. Now, let's look at what is called as the Y-linked genes. Y-linked genes are the genes that are present on the non-homologous region of the Y chromosome. So these genes are Y-linked. These are Hollandric genes and they are only present in males. Example for the same is hypertrichosis, that is the excessive growth of hair on the pinna of the ear. So my dear students, in this module we have discussed about crossing over and sex-linked inheritance. In the next module we would be discussing about color blindness.